Welcome to G Fun Facts Online. Today, we're diving into one of the most unexpected and revolutionary stories in modern medicine. What if the key to saving the brain from diseases like Alzheimer's and Parkinson's was hidden inside the very drugs designed to kill cancer? It sounds like a paradox. Cancer is a disease of uncontrolled cell growth, while neurodegeneration is a disease of devastating cell loss. They seem like polar opposites. Yet, scientists are discovering that these two formidable enemies share a secret battleground deep inside our cells. This has sparked a groundbreaking strategy, taking powerful cancer drugs and repurposing them, turning weapons of cellular destruction into tools of neural protection. This isn't just a clever shortcut. It's a paradigm shift born from a deeper understanding of biology, one that could dramatically accelerate the search for treatments for some of our most feared diseases. Today, we explore this unlikely alliance. First, let's understand the shared battleground. How can two such different diseases share common ground? It turns out that fundamental processes that keep our cells alive and healthy can fail in different ways, leading to either cancer or neurodegeneration. Think of it as the same system breaking down, but with two different outcomes. One of the most critical systems is called autophagy. This is essentially the cell's internal recycling and garbage disposal program. When proteins get damaged or organelles wear out, autophagy engulfs this cellular junk and breaks it down for reuse. It's crucial for keeping cells clean and functional. In neurodegenerative diseases like Alzheimer's and Parkinson's, this system often becomes sluggish and inefficient. The cellular garbage disposal gets clogged. Toxic misfolded proteins like amyloid beta, tau, and alpha-synuclein start to pile up. This buildup disrupts cell function and ultimately kills neurons. So for these diseases, the goal is to boost autophagy to get the cleanup crew working overtime. In cancer, the story is flipped. Cancer cells are masters of survival. In the harsh, low-nutrient environment of a tumor, they hijack autophagy and crank it into high gear. This allows them to recycle their own parts for energy and building blocks, helping them endure conditions that would kill normal cells. So while some cancer drugs try to inhibit autophagy, others, when used differently, can actually enhance it. And that's where the opportunity lies. Cancer drugs known as tyrosine kinase inhibitors, or TKIs, like nilotinib and bosutinib, have been found to stimulate this cellular cleanup process, offering a potential way to clear the toxic junk from ailing neurons. Another common thread is protein misfolding. Proteins are the workhorses of the cell, and they need to be folded into precise 3D shapes to function. When this folding goes wrong, you get toxic sticky clumps. In neurodegeneration, this is the central problem: amyloid plaques and tau tangles in Alzheimer's, or Lewy bodies in Parkinson's. These aggregates are directly toxic to brain cells. But protein aggregation also happens in cancer. The famous tumor suppressor gene, p53, which is mutated in many cancers, can create a protein that misfolds and clumps together. These clumps not only lose their ability to stop tumors, but can even gain a new toxic function that helps the cancer progress. So any strategy that can prevent or clear these protein aggregates could be a win for both fields. Then there's the strange phenomenon of cellular senescence, which gives rise to what scientists call zombie cells. Senescence is a state where a cell permanently stops dividing, often in response to damage. This is a powerful anti-cancer defense, preventing damaged cells from becoming tumors. Many cancer therapies, like chemotherapy, work by forcing tumor cells into this state. But there's a dark side. As we age, these non-dividing zombie cells can accumulate in our tissues, including the brain. They don't just sit there quietly. They spew out a cocktail of inflammatory chemicals that damage their healthy neighbors. In the brain, this chronic inflammation is a major driver of neurodegeneration. This has led to an exciting new class of drugs called senolytics, which are designed to selectively hunt down and destroy these zombie cells. And many of the first senolytics are repurposed cancer drugs, like the TKI dasatinib, which has shown promise in animal models for clearing senescent cells and improving cognitive function. Finally, let's consider neuroinflammation. A little bit of inflammation is good. It's the body's response to injury. But in diseases like Alzheimer's and Parkinson's, the brain's immune cells become chronically activated, creating a state of constant, low-grade inflammation. This smoldering fire is incredibly destructive to neurons. Cancer also has a complex relationship with inflammation. In fact, tumor-promoting inflammation is a recognized hallmark of the disease. This overlap means that anti-inflammatory cancer drugs could potentially be repurposed to quench the fire in the brain that fuels neurodegenerative decline. So this shared biology isn't just theoretical. It has produced a real pipeline of promising cancer drugs being tested for a new mission. Let's look at the arsenal. The tyrosine kinase inhibitors, or TKIs, have been leading the charge. These drugs are a cornerstone of cancer therapy, designed to turn off the switches that tell cancer cells to grow. Nilotinib and bosutinib, originally for leukemia, were repurposed for Parkinson's disease. The idea was to boost autophagy and help neurons clear out toxic alpha-synuclein. Early trials were promising, showing the drugs were safe and could enter the brain. However, larger, more recent trials for nilotinib have unfortunately been disappointing, showing no clear clinical benefit. This is a common story in science. What works in the lab doesn't always translate to patients. But these trials provided invaluable lessons, and a recent trial for a similar drug, bosutinib, in patients with Lewy body dementia, did show it could reduce toxic proteins and improve daily living activities, suggesting it warrants more study. Another TKI, masitinib, has shown significant success in a large clinical trial for ALS. When added to the standard treatment, it significantly slowed the rate of functional decline in patients and is now under review for approval in Europe. This could be a major breakthrough for a devastating disease. A more recent and very exciting development came from a powerful computational approach. Researchers analyzed gene activity in single brain cells from Alzheimer's patients to create a disease signature. Then, they searched a massive database for existing drugs that could reverse that signature. They found a surprising pair of cancer drugs, letrozole, used for breast cancer, and irinotecan, used for colon cancer. Their hypothesis was that letrozole would help neurons, while irinotecan would calm down the inflammatory glial cells. In a mouse model of Alzheimer's, this combination was a stunning success. It reduced amyloid and tau, prevented brain degeneration, and restored memory. Human clinical trials are expected to follow soon. Of course, not every attempt works out. The story of bexarotene is a cautionary tale. This skin cancer drug caused a huge wave of excitement back in 2012, when a study reported it could rapidly clear amyloid plaques in mice. But in human trials, it failed to reduce amyloid in the brain and caused risky side effects. It turned out the drug just couldn't get into the brain in sufficient amounts. It highlights the critical need for rigorous clinical validation. One of the most promising avenues targets the internal skeleton of the neuron. Our brain cells have a network of microtubules, which act like highways for transporting essential cargo. In Alzheimer's, the tau protein, which normally stabilizes these highways, becomes abnormal and clumps together, causing the whole system to collapse. Enter epothiolone D, a microtubule stabilizing drug used in chemotherapy. The idea is that it could shore up these highways, compensating for the defective tau. In mouse models, it has been shown to improve this transport system and rescue cognitive deficits. A human clinical trial has been completed, and the neuroscience community is eagerly awaiting the results. However, all these brilliant ideas face one colossal obstacle: the blood-brain barrier. This is a protective, highly selective membrane that lines the blood vessels of the brain. Its job is to keep out toxins and pathogens. The problem is, it's so good at its job that it also blocks more than 98% of potential medicines. For a cancer drug to work on a neuron, it has to get past this fortress. For decades, this has been the great wall of neurotherapeutics. But now, incredible new technologies are emerging to breach it. One of the most futuristic is MRI-guided focused ultrasound, or FUS. This is a non-invasive procedure where a patient wears a helmet containing over 1,000 ultrasound transducers. First, they receive an injection of harmless microbubbles. Then, the helmet focuses sound waves on a precise target in the brain. The sound waves make the microbubbles vibrate, and this mechanical jostling gently and temporarily pries open the junctions of the blood-brain barrier. It creates a window, lasting a few hours, for drugs to pass through before the barrier safely reseals. Clinical trials are already using FUS to deliver therapies for Alzheimer's, Parkinson's, and ALS. Another approach is nanotechnology's Trojan horse strategy. This involves packing drugs inside engineered nanoparticles. These tiny particles can be coated with molecules that trick the blood-brain barrier into carrying them across. Once inside the brain, they can release their therapeutic cargo in a controlled way. So, how do scientists even find these candidate drugs in the first place? It's a systematic hunt driven by big data and artificial intelligence. One key tool is the connectivity map, a huge database of how thousands of drugs affect gene activity. Scientists create a genetic signature of a disease, like Alzheimer's, by seeing which genes are turned up or down. Then they use AI to search the database for a drug that produces the opposite signature, one that can theoretically reverse the disease at a genetic level. This is exactly how the letrozole and irinotecan combination was discovered. It's a bit like finding the perfect key for a complex lock, but on a massive, computational scale. These predictions are then validated in the lab using high-throughput screening, where robots rapidly test thousands of compounds on cells to see if they have the desired effect, like protecting neurons from a toxin. Despite all this promise, the road to repurposing is filled with hurdles. Cancer drugs are toxic by design. The acceptable level of side effects for a terminal cancer patient is very different from what's acceptable for someone with a chronic, slowly progressing neurodegenerative disease. Doses often need to be lowered, but then the question becomes, is the drug still effective? The biggest challenge, however, is economic. Many of these promising candidates are older, generic drugs. Pharmaceutical companies have very little financial incentive to spend hundreds of millions of dollars on new clinical trials for a drug they can no longer patent. This funding gap, often called the Valley of Death, means that many promising ideas never get tested in humans. It's a market failure that often leaves the heavy lifting to underfunded academic groups and nonprofits. New models are being explored to solve this, but it remains a major roadblock. In conclusion, the convergence of cancer research and neuroscience represents a true revolution. By understanding the deep biological connections between cell proliferation and cell death, we are unlocking an entirely new arsenal in the fight against neurodegeneration. This strategy leverages decades of investment in oncology and harnesses the power of artificial intelligence and big data to find new hope in old medicines. The path is challenging, littered with scientific and economic obstacles, but the progress is undeniable. This unlikely alliance is accelerating the quest for therapies that could one day slow, stop, or even reverse these devastating diseases. For the millions of people affected by Alzheimer's, Parkinson's, and ALS, this paradigm shift offers a renewed and powerful sense of hope for the future. Thanks for tuning in to G Fun Facts Online.